as energy sources, but are also used to make other compounds that the body needs to function properly. So we know that we already know about the digestion of fats, namely triglycerides. They're broken down, and these are the best bang for the buck in terms of energy sources for the body, and we're going to talk about how in this lecture. But we know that here's a picture, of course, of the triglyceride molecule, like glycerol backbone, and the 3 fatty acids. So we know triglycerides are broken into smaller molecules by bile salts. Then they're acted on by lipase enzymes to break them down into the glycerols and monoacyl, to fatty acids and monoacyl glycerols. Then those recombine once they cross into the body, and then they're brought to the different tissues in order to be used for energy or other purposes. And so, with lipid metabolism, there are 5 different types of lipid metabolism. We're going to talk about 3 types today. They include beta oxidation, which is the breakdown of triglycerides. We're going to talk about lipogenesis. We're going to talk about ketogenesis, and in the next block, we're going to talk about cholesterol synthesis and how that pathway works. But then some of the other lipids that are metabolized, for example, we have phospholipids. Those are metabolized, or those are synthesized by substrates of the glycolytic pathway, namely glycerol 3-phosphate. We're going to spend most time talking about that. So interesting about all of the lipogenic or pathways is that they all either use or end up with acetyl-CoA. We're going to talk about 2 examples of that today. So when are lipids used as a source of energy? This occurs when glycogen stores have been depleted from the liver and we've already used all of our blood glucose. So then that's when we transition to using lipids as our primary energy source. This process is initiated by the hormones glucagon and epinephrine. They're secreted into the bloodstream, which triggers the lipase enzymes to break down triacylglycerols into that glycerol backbone and fatty acids to be used as energy sources. So, in terms of glycerol, as you can see the structure at the bottom, it becomes first dihydroxyacetone. We know that as intermediate of glycolysis. And then, of course, as was talked about earlier, the dihydroxyacetone must be converted to glyceraldehyde 3-phosphate in order for it to continue down the glycolytic pathway or to be used in gluconeogenesis. Any questions so far? Okay, so look at there are only 2 pathways left. Y'all are rocking with these pathways. I'm gonna have a lot of questions for you in about 2 weeks about the pathways. I'm sure you're gonna do very well. So we're gonna talk about beta oxidation and lipogenesis. And as you can see, beta oxidation is the breakdown of those fatty acids to acetyl-CoA, which will be used in the TCA cycle for energy production. And fatty acid synthesis will convert that excess glucose first into acetyl-CoA, and then if it doesn't need the acetyl-CoA for energy purposes, then you will look like me and you will have excess fat stored in the adipose tissues. All right, beta oxidation. So beta oxidation occurs, as you can see, here's a fatty acid here. You have the alkyl group and the carboxylate and circled in green. Beta oxidation, its name comes from the fact that the bond between the beta and alpha carbon is broken, in two carbon molecule segments to create acetyl-CoA. So the bond is broken here. This is the beta carbon, the alpha carbon. The bond is broken here. This molecule becomes acetyl-CoA. And this is done in a two-step process until all you have are acetyl-CoA molecules. So you guys don't need to know these steps in this pathway, but just wanted to show exactly what was going on. So in beta oxidation, as I mentioned, triglycerides are broken down into fatty acids. Those fatty acids are then broken down into acetyl-CoA, 2 carbons at a time. To get one acetyl-CoA, it's a 4-step reaction shown here. This green horizontal line represents the bond between the beta and alpha carbon. What happens is that beta carbon, it undergoes 4 different modifications, resulting in the cleavage of acetyl-CoA in the final step shown here. And this 4-step reaction produces 1 acetyl-CoA. So if you have a 16 carbon sugar palmitate, this reaction will occur 7 times to produce 8 acetyl-CoA. As you can see here, in this 4-step process, you produce 1 FADH2, 1 NADH2. So if you have a 16 carbon fatty acid, you will get 8 acetyl-CoAs, 7 FADH2, and 7 NADH2, which of course we know those will produce a significant amount of ATP, much more than we saw in the glycolytic pathway. So as I mentioned, this process removes beta oxidation, removes carbons, 2 carbons at a time to create acetyl-CoA. That's the first step shown here. The second step, you guys are very familiar with this, those acetyl-CoAs are brought into the TCA cycle, then the electron transport chain to produce ATP. Any questions? All right. So before beta oxidation can occur, the fatty acid must be activated. That's the initial signal or trigger for beta oxidation to occur. And that uses ATP to add that famous CoA group to the fatty acid molecule. And this uses one ATP molecule, but because it is breaking 2 of the phosphate bonds, typically when we see ATP use it, it's broken to ADP. But in this case, it goes from ATP to AMP. So that, because 2 phosphate bonds are broken off of the ATP, that's kind of equivalent to 2 ATP molecules being used. So this is basically a high energy reaction. It's irreversible, and this is going to signal the body, okay, now we need to do something with this fatty acyl-CoA group in order to break it down further. And so what must be done, all of these steps have occurred in the cytosol of the cell. In order for beta oxidation to occur, we need to bring this fatty acyl-CoA group into the mitochondria. So what happens is the fatty acid and that CoA, the fatty acyl-CoA group, must add a carnitine moiety to it. That carnitine moiety will enable the fatty acid to be transported into the mitochondria. This is through the carnitine shuttle system. So the first enzyme in this step is catalyzed by carnitine acyltransferase 1. This is the rate-limiting enzyme. What that does is the fatty acyl-CoA carnitine is added to it, removing the CoA, making fatty acylcarnitine. That fatty acylcarnitine can then be transported through the carnitine transport protein into the mitochondria, where carnitine acyltransferase 2 enzyme will catalyze the reverse reaction, removing the carnitine, adding back the CoA, And once the fatty acyl-CoA is in the mitochondria, that will begin beta oxidation, where 2 carbons will be removed at a time. And here are the steps to this pathway that I just mentioned. All right, any questions about this pathway? Yes?
The acetyl-CoA like precursor, that's inside the matrix that kind of gets put back onto the fatty acid.
This one?
Yeah, it's just sitting there in the
Yes.
matrix, or it also gets transferred through.
Yeah,
from
it's
the
this.
outside
Some of that stuff is already there, and I can't remember if this is one of the examples. Some of the cells, particularly the mitochondria, has reagents in there that they can't transport out on their own, so they're just staying there for the uses in these reactions, and so they're kind of like waiting to be used and reused, if that makes sense. So, but this one is just hanging out there. It knows what it's going to be used for, and then once the reaction is done, it's released back into the mitochondrial matrix for another reaction to occur with another fatty acid. Yes.
So it looks like the end product is the same thing, the carnitine and the fatty acyl carnitine. Why it has to be transported in and out just for it to be put back together?
Because it's too big to go through the mitochondria on its own. So that carnitine group makes it allows it to be transported. This carnitine group that's added allows this fatty acyl to be transported into the carnitine shuttle. It's like kind of escort, I guess you could say. Without the escort, it's not going to be able to cross into the mitochondria. Yes.
Does the transport protein in the middle, does that move anything? I see the one on the right, pushes the fatty acyl protein in the matrix, but in this process, does that middle transport protein have anything to do?
This one.
The one on the.
I think it pushes the carnitine back out.
Yeah.
This moves that carnitine group back out.
Okay.
Yeah. So this takes the fatty acyl whole carnitine group in, and this will split that. This enzyme will split the fatty acyl and the carnitine, add the CoA back, and will move that carnitine group back out. Yes.
Big picture, this is just trying to get that fatty acyl into the mitochondria to be
100%. And it can't
That's all
do it
that
on
is.
its own. Yeah. Any other questions? So this is just to show, this is the same diagram before, it's rotated 90 degrees. So in this diagram, you see that there's only one membrane, but you guys all know that the mitochondria has an outer membrane and an inner membrane. Same thing is happening, it's just that shuttle is transporting from the outer membrane to the inner membrane space into the matrix. So that's all. So as we've seen with other conditions in the pathways that we've talked about, if there is any deficiency or altered protein, be it just a protein or an enzyme, that's going to lead to a disorder. In this case, some of the common beta oxidation disorders are systemic primary carnitine deficiency, and what this condition is basically that carnitine transport protein is deficient, so you cannot get fatty acids into the mitochondria. So that's going to lead to, of course, muscle weakening because your muscles they don't have enough energy source because the fatty acids are not going to be broken down to produce ATP. That's the hypotonia. Then you also have hypoketotic hypoglycemia. So we already know that fatty acid oxidation occurs when glycogen stores have been depleted and blood glucose is depleted. So we're already at a hypoglycemic state in order for fatty acid breakdown to occur. Now, what this hypoketosis is, under normal conditions, we're going to talk about ketone body or ketogenesis a few slides forward, but I'm going to give you a preview to this now. So normally, when the body undergoes, when you're in particularly starvation and it switches to fat metabolism, if the metabolic proteins are working properly, that those fatty acids will be broken down. They will increase the amount of acetyl-CoA that's produced. When there's a lot of acetyl-CoA, those acetyl-CoAs will combine to produce ketone bodies. So our bodies have basal levels of ketone bodies, but in this case, in systemic primary carnitine deficiency, we're hypoglycemic already, but ketone bodies cannot be generated because we cannot metabolize the fat because it cannot enter into the mitochondria. So you have what is called hypoketotic hypoglycemia. Does that make sense? Any questions about that? So the treatment for that, we can buy this at GNC or Walmart anywhere, is L-carnitine, and that is just that. It helps get those fatty acids through the mitochondria into the matrix in order to be broken down. So the other condition is the myopathic CAT2 deficiency, and this, we know that CAT2 enzyme is found in the mitochondria. And what happens when this enzyme is deficient, again, you're going to see that muscle weakness, and you're also going to see muscle wasting and breakdown, and this is due to the muscle's not getting any energy sources, and so what's going to happen is that myoglobin in the muscle is going to break down because the muscle tissues are not getting any energy source. That breakdown of myoglobin is going to spill into the urine, and that's why you're going to see the myoglobinuria, and so that also is going to cause increased triglycerides in the muscle tissue, which contributes to the muscle dysfunction as well. And so how you would treat this, you would tell your patients to, basically they would have to have a diet low in fats and proteins, and you would want them to eat high carbs, so that becomes a main energy source they use instead of fats. Does that make sense? Yes.
In regards to the first problem, if someone comes to us and says they're on like keto diet, do we recommend them taking L-carnitine as a supplement to help?
Yeah, you can definitely, but it depends on how long they've been on it too, because and that's a great prelude to what I'm going to talk about, because I was going to ask you guys are all young, fit, and all that stuff. I was going to ask you, have any of you used a keto diet for any reason, and if so, do you mind sharing? Oh, yes. Sorry. Yes.
I haven't, but my dad was showing pre-diabetic symptoms. He switched to a keto diet, and it completely went away.
Nice, perfect. Anybody else? Did you raise your hand?
I did it back in the heyday, like 2010, 2011, 2012. I lost a lot of weight, but my triglyceride levels were pretty high. Naturally, HDL was high, LDL was low. It took me about 3 weeks to acclimate.
Okay.
But I think what I found was that it was very difficult socially.
Because there's carbs and everything is carbs. Yeah, definitely.
I became a drink guy.
Yeah, as many times as I've tried to do the keto diet, I like to eat. So I can't go out to eat because I'm gonna want some bread, I'm gonna want some fries, all that stuff. Y'all can see that. But yeah, so 2 great examples of using a ketone diet. I've known some other people to use it for one of the previous classes. There was a competitive bodybuilder. She used it, of course, to get grades. And so I've tried to use these ketone strips, which tells you the amount of ketone bodies you have, but I ate too many carbs, so never really got to the ketogenic level that I wanted to. Was there somebody else? I saw somebody else was there. Yes.
Is L-carnitine something that you get from your diet, or does your body make that?
You can get it from the diet. I'm not sure what foods have it, but it's a dietary supplement that you can buy. I don't know. That's a good question. I don't know if it's something that the body makes a lot of. I'm sure it probably makes it, but I'll find out for you. Any other questions? Yes.
Speaking of produced in liver
Thank you. It's produced in liver and kidneys. Appreciate it. Any other questions? All right. So, as I mentioned, ketone bodies are formed when we break down fatty acids, and what happens is when we get ketogenesis or excess ketone bodies, that means that basically we have more acetyl-CoA produced than we can use. So that means that the TCA cycle or the gluconeogenic cycle, any pathway that needs to the energy source, be it the form of glucose or ATP, those are kind of backed up. So though that excess acetyl-CoA that's produced is going to combine to form three main ketone bodies. Those are acetate, that's one example. Beta hydroxybutyrate is another ketone body, and then acetone are ketone bodies that are found in the body as well. And so we normally have a very low level, and when we go on a keto diet, it increases. But we're going to talk about a condition where it can be very severe and life-threatening. And so, because ketone bodies are acidic, they will lower our body's pH. Remember, the basal range is between 7.35 and 7.45 for our body. And so, if we have excess ketone bodies, it will lower it far below that. And we find that, of course, during starvation, our body will transition to using lipids to metabolize because the sugars are gone. Diabetes, of course, is another condition where ketogenesis occurs. And however, ketosis and diabetic ketoacidosis are very different things. And so we're going to talk about, we know in diabetes, insulin doesn't work properly. This picture here kind of demonstrates what's going on. So when our blood glucose is high, insulin will be released, causing blood glucose to be lowered, converting glucose to glycogen in the liver. When blood glucose is low, glycogen will be released to break down that glycogen into glucose, which will put glucose into the blood. And so in diabetes, insulin does not work properly. And so what happens is in this diabetic ketoacidosis, insulin doesn't work, so glucose is not getting in the tissue to become an energy source. We have excess lipid breakdown, but that excess glucose in the blood is going to spill over into the urine, taking electrolytes with it. So remember we talked about that polyphagia, polydipsia, polyuria, that's going to occur. But then you're going to have a significant ketone body production because fats are going to just be broken down to try to be used as energy. But what's going to happen is your blood pH is going to drop significantly. And we know that acidic environments where it's not supposed to be acid in our body is going to cause a number of conditions. And so this person, a person that undergoes severe diabetic ketoacidosis, would need to be brought to the hospital immediately, and dialysis needs to occur to get that, get that glucose and the ketone bodies out of the blood in order to return the blood to the normal pH range. Any questions about that? Yes.
So what really is the difference between the diet-induced ketosis and then diabetic ketoacidosis? Technically, couldn't both of them become ketoacidosis and dangerous-free?
No, diabetes is going to be worse because diabetes, it's a number of things going on, and that excess glucose floating in the blood is going to cause a lot of problems too. And I don't think the lipid breakdown in diabetic ketoacidosis, I think that lipid breakdown, it just won't stop. So the ketone bodies produced are going to be far greater than what can be produced in a normal ketotic state where we have low carb, high fat diet. So that makes sense. Any other questions? Yes.
So I've had a lot of students try to get through diet, especially when you talk about MCT.
Trust me. Yeah, I know.
At what point should you make that recommendation? Hey, it's time for you to go back to a regular.
I was gonna say when they start fighting people because they're in a bad mood, but no. Because that diet, I mean, I don't know about you guys that have been on it, but that diet made me very irritable when I tried to do it. And I still didn't do it. But I would recommend that you, like, if they're not seeing were they seeing results? Maybe they could like figure out how to do it in a shorter window. You mean because you're trying to figure out when to tell them to go back to the normal diet?
At what point are you starting to have like these increased
Oh, I mean, it can start a few days after you start if you're very diligent, your body can go into ketosis in a few days. But I don't know the exact timeline, but I know some people that have been on this diet for 3 and 6 months, and the people that I know, they didn't have any significant health issues. So, like in terms of like other conditions, like they were typically healthy people. Like one of my colleagues, when I joined the Army, we were office mates, and she was forever on a ketose diet. She's the one that got me using the strips. They worked for her all the time, but never for me. She was very diligent, and she did it like off and on for the 3 years that we worked together. She would do it for like 3 and 6 month periods, because she was going to Miami or whatever, Understand? I mean, I get it. Like you want to look your best when you're around people, So, but I looked how I looked when I was around. But seriously, I would tell them that like if it's working for them and they're not noticing any other conditions, and I'm sure your soldiers are kind of that, right? So they probably are typically like very healthy. They might not even realize they're having some side effects, but if they're not experiencing like any other symptoms, I would tell them, hey, stay on it as long as you need, but after height and weight, they can eat real food sometimes. But if it's a persistent problem like they're constantly on ADCP program, they might need to see a nutritionist or something. Any other questions? All right. Now we're going to talk about fatty acid synthesis or lipogenesis. Lipogenesis occurs when we now have excess acetyl-CoA that has been produced, and we're going to make fatty acids. And this is also excess acetyl-CoA and our glycogen stores. So we already have enough glycogen, so that excess glucose that comes into the body will be first converted to acetyl-CoA and then undergo the lipogenic pathway to produce up to 16 carbon length fatty acids, which we know is palmitic acid. This process is initiated by the acetyl-CoA interacting with the acyl carrier protein shown at the bottom of the screen. That is the signal for lipogenesis to occur. So lipogenesis is not the opposite of beta oxidation. It's a different pathway entirely. Instead of gaining FADH2 and NADH, this process uses NADPH. And so in this pathway, it's activated by insulin that's going to bring the glucose into the tissues, and that excess glucose will be converted into fatty acids. And so the first step, once it's been activated, once that acetyl-CoA has been activated by that ACP group, N-acetyl-CoA will combine with a 3-carbon malonyl unit to produce, of course, a 5-carbon intermediate. In that 5-carbon intermediate, the next reaction will remove a carbon dioxide, so you have 4 carbons, and then subsequently 2 carbon groups will be added on until we get to 16 carbon fatty acid. Any questions? So here are the primary differences between beta oxidation and lipogenic pathways in terms of where they occur, the starting substrate, starting coenzymes, and the final products. You guys don't need to worry about the types of reactions used, but I would know where they occur, what the final and initial substrates are, and what coenzymes are used. Any questions about this chart? All right. So now we're going to talk about how proteins are used in these pathways. So proteins, of course, are broken down into amino acids, and they can also be used as energy sources, as an energy source. So we know proteins are broken down first into polypeptides in the stomach, and then into amino acids in the small intestines, and then they're absorbed into the body for further use. So our body maintains a protein balance, so there's protein turnover. And so what happens is when we consume protein in our diet, they're broken down to amino acids. Those amino acids can go to be used to make other molecules that the body needs, such as nucleic acids, amino alcohols, of course, other proteins and enzymes. However, of course, those amino acids can also, their carbon skeletons can be used and will serve as intermediates for all of the pathways that we've talked about so far, including the TCA cycle, acetyl-CoA production, and also in the gluconeogenic pathways as well. And so when we have excess, when we eat too many proteins, since our body needs to maintain a protein balance, the body will remove that excess protein in the form of ammonia, which will be excreted in our urine as urine. And that is how the body maintains its nitrogen balance. So too much nitrogen has to be removed in the form of urea. And we know there are cases when we don't eat enough proteins, and in the most severe cases, one of them being anorexia, that's very dangerous because now your muscles are going to be breaking down, and then that's going to affect your heart, and your heart's not going to be able to work properly, nor is it going to be able to pump blood needed throughout your body for energy production. So that's very dangerous, and that's an example of severe malnutrition and protein breakdown. So when we have too much nitrogen, when we eat too many dietary proteins, and this is something I'm very good at, because I like to go to Brazilian steakhouses. I eat a lot of meat. I like almost all the different kinds. And I've been to almost all the Brazilian steakhouses here many times, and I just moved here. Truthfully, I lived in Germany for 6 years, and every time I came back to visit my parents, they already knew what was up. We would go the day I got back, take a shower, be sleepy, jet lag, fuck with a child. Tamagotchi, yeah. And I actually went, that's where my mom wanted to go for Mother's Day, so she doesn't even like that place, but she was overruled by the rest of us, so sorry, Mom, but we went for Mother's Day. She does like the salad bar. Let me not make you guys think I'm a horrible person. She does love the salad bar, but it was a win-win. So when we have too much protein, we eat too much protein, our body cannot use that protein, so it's going to be removed in the form of ammonia, but it will use the carbon skeletons for other purposes. So the first step in removing that ammonia group, either to make another amino acid or to get it removed from the body, is a transamination reaction. And so in this reaction, you have the alpha amino group will be removed from an amino acid, typically alpha ketoglutarate. And in this reaction shown here, we have alanine and alpha ketoglutarate. Where have we seen alpha ketoglutarate? Citric acid cycle. Perfect. That's the intermediate citric acid cycle. So what happens is they will interact. The transamination reaction will occur by alanine transaminase to produce pyruvate. Pyruvate is from what pathway? Glycolysis. And then that produces glutamate. Glutamate is a neurotransmitter that's needed in the body. So this process right here is one way our body produces non-essential amino acids, as you can see, because alanine and it's a reversible reaction. Alanine and glutamate are both non-essential amino acids. So, but if we have excess nitrogen, this glutamate will now undergo oxidative deamination. So here's that reaction from the previous slide. That glutamate will now, within the presence of glutamate dehydrogenase, produce alpha ketoglutarate again, and that extra ammonium group, which will then be removed from the body via the final pathway we're going to talk about in this block, which is the urea cycle. And we've already talked about like how amino acids and, well, we're going to talk a little bit about how amino acids undergo the ketogenic pathway, just a small snippet, but the urea cycle is the way the body removes excess ammonia from the body. And so it converts that ammonia group into urea. Urea are transported. This reaction occurs in the liver. The urea is transported to the kidneys in order for excretion as urine from the body. So this reaction occurs, as I mentioned, in the liver, in both the mitochondria, as shown by the darkened box, and then the cytosol. So what happens is that ammonium group will interact with carbon dioxide in the presence of carbonyl phosphate synthetase 1 to produce carbonyl phosphate. Carbonyl phosphate will interact with the ornithine transcarbamylase enzyme to produce citrulline. Citrulline then will leave the mitochondria, enter into the cytosol of the liver, interact with ATP and aspartate to produce arginosuccinate. Arginosuccinate will then become arginine, releasing fumarate. Where are we seeing fumarate? The TCA cycle, right. So then that arginine will then be converted to urea, which will travel to the kidney. So this won't be a surprise to you, but there's going to be a deficiency, of course, of one of these enzymes. And when there is, particularly ornithine transcarbamylase, that is an X-linked recessive condition where the ornithine transcarbamylase is deficient. And so what happens is carbonyl phosphate will build up, it will become orotic acid. Orotic acid is very toxic, and that is harmful to the liver. And of course, because we cannot convert the ammonia into urea, we're going to have hyperammonemia. I did not mention, but this is very important, that carbonyl phosphate synthetase 1 is the rate limiting reaction in this pathway, the urea cycle. So what happens in OTD deficiency, of course, ornithine transcarbamylase, citrulline, will be decreased, carbonyl phosphate will increase, and it will be converted to uric acid, which is toxic. And then we will have hyperammonemia. That hyperammonemia will lead to symptoms such as positive asteriscus and cerebral edema. What's happening is that positive asteriscus is a test where you're looking at a person's motor function. The hyperammonemia interacts and disrupts the motor systems in the brain. And so when you are examining your patient, is when you try to tell him to make his hands as a 90 degrees, they're going to be flapping because the motor centers are messed up, and it's disrupting like the neurotransmitter imbalance in the brain. Also, cerebral edema will occur, and this is because the hyperammonia, that ammonia is able to enter the brain easily, and that excess ammonia will be broken down in the astrocytes to glutamine. Glutamine likes to, it's osmotically regulated, so glutamine will cause water to be drawn into the astrocytes. That's going to cause brain swelling, and brain swelling is bad, and that can lead to all the symptoms shown here in this picture. And so the treatment for OTD deficiency are two drugs that act primarily on the digestive tract, bacterial gut. What they both do together, they both basically stop the ammonia from being absorbed into the body. So that ammonia that is produced by the breakdown of proteins will be excreted. It won't be absorbed, and it will be excreted in our feces. And so that's going to stop ammonia from being absorbed into the body and causing more effects. And so these two drugs are lactulose and rifaximin. Are there any questions about the urea cycle? Yes. Carbonyl phosphatase synthetase 1. Any other questions? All right, and this is just a summary of the urea cycle. Oops. So this pathway is removing excess nitrogen from the body. So now we're going to talk about how amino acids are also, we've kind of alluded to this, that they can be used for a number of purposes, particularly as it relates to the pathways that we talked about in this block. Their carbon skeletons can be used to produce several intermediates in the glycolytic, gluconeogenic, and TCA cycles as shown here. So typically, 3 carbon amino acids will be converted to pyruvate, 5 carbon amino acids will be converted to this 5 carbon alkyl glutarate, and 4 carbon amino acids, that skeleton, will be converted to oxaloacetate. Typically speaking, that's, of course, there are exceptions to this. And so, also shown, the pink or salmon-colored boxes represent amino acids that are thought to be glucogenic, and the yellow boxes represent amino acids that are said to be ketogenic. You see, because the ketogenic ones will form ketone bodies, of course, or acetyl-CoA, which we know is the substrate for ketogenesis. And then these other molecules operate in the glucose generating pathways, typically starting from glucose. And so, as I mentioned, amino acids, the 3 carbon skeletons will likely be converted to pyruvate. 4 carbon skeletons will be converted to oxaloacetate, and the 5 carbon skeletons will be converted to alkyl glutarate. And so these glucogenic carbon skeletons. They're said to be glucogenic if they produce pyruvate, ketoglutarate, succinyl-CoA, any of the substrates in glycolysis in the TCA cycle. The ketogenic skeletons from amino acids produce acetyl-CoA or ketone bodies. Any questions? All right, you guys take a 10-minute break. You have another class starting at 10?
Yeah.
Oh, guys, I'm sorry. I didn't know. I thought the whole schedule was pushed back.
Even if he makes good son, never because he has enough electric box to make the time. Well, in the sense that he's given enough electric box to get yeah. Because I mean, it's a lot more content. Yeah, but then like he already everyone is good at
It might not be I'm not sure
questions. I'm so excited to see the last question was, you guys wanted more, right?
I like this.
He is good. Great.
You still gotta study.
Yes. But I don't know.
Yeah.
She takes a slide from every 3 or 4 seconds. Okay, make sure you move off the side. Next one, make sure this.
Yeah, but I forgot those other 3 slides.
I better put it back. Make sure you guys study. Oh, you have a question? I'll make sure you go back to it. Oh, it's Daddy's new content right now.
Yeah.
Yeah, very intuitive. Remember all other answers are the different. You should know why they're different.
Yeah. I'll look into that to see. I'm not sure. Like I said, I'm not sure. I'll look into that.
Oh, no.
Like, for example, so for transamination, what's the transamination?
Yeah.
I'm just like, we have time. Like, this is the last time we get 2 weeks of like priorities right now.
Yeah.
Like, we have a 4 day before the AP2 and then, so like, we have time.
Substrates and main enzymes.
No, it's Monday. It's the 30th. The Monday coming back at the end of June. No, after that. Coming back by 4 day, you go straight to AP1 and then the end of it. No. Yeah, that was the 4 day. Yeah. But right now it's just like micro AP1 and switch to AP2, slide of him, slide of AP1, switch back to AP1. Friday. Yeah, but then a virtual weekend. You can do it. Putting microbiome. Yeah.
So this is last time we had on Friday night. Yes.
It's not the least of like it's the first evolution. I know if you're not first
Yeah, lipogenesis is one of the facts. So we didn't talk about that. It's probably not
Everyone's
like, it's easy.
the old guy. Because I know it is by like
Good
yeah.
question.
Which I'm not complaining about. Is it a lot of share? So yeah, you don't want to get happy.
You don't have to tell me.
I'm gonna ask you where the great intersection line where the size 22. Make sure you point out.
You know what I mean? You guys have class. Oh, still.
Here.
Yeah. You only have 200 slides. You said that's right.
I think the biggest difference is about
Yeah. Yeah, I saw that shit. I'd rather make big controversy. What? No, there was like a bunch of groups in the state.
slide. So, any questions about what we talked about this far? No questions? Comments, concerns? All right, so synthesis of amino acids. Now, we've already talked about this a little bit in terms of that transamination reaction that we talked about earlier. So we know that that non-essential amino acids are made from amino acids that we already have in the body. Via transamination, we saw that alanine was converted to glutamate. Those are both non-essential amino acids. However, as we know, the essential amino acids we must eat in our diet. And so, how we make non-essential amino acids, that alpha keto acid carbon skeleton attained from the pathways that we discussed, the TCA cycle and glycolysis, are converted to amino acids by the transamination reaction. So, in glycolysis, 3-phosphoglycerate and pyruvate can be used to make serine, alanine, and which can be converted to glycine and cysteine. And from the TCA cycle, oxaloacetate and alpha-ketoglutarate, that those carbon skeletons will undergo a transamination reaction to produce the rest of the non-essential amino acids: aspartate, glutamate, asparagine, glutamine, proline, and arginine. And so all this is showing you is that you guys don't need to remember this, but this is just showing you that the amino acids, the pathways kind of go backwards and forwards. They're reversible. So we saw previously how amino acid, their carbon skeleton were used to make intermediates of these pathways. Now these intermediates can go back opposite direction to produce non-essential amino acids. Does that make sense? So we've talked about both catabolic and anabolic pathways in this block. You know, catabolic, breaking down, it's used to produce energy. Anabolic, building up, often require energy to build up and store molecules. And so here is an overview of pretty much everything we've talked about in this block, how the macronutrients, proteins, polysaccharides, and lipids are broken down and how they're used in this central metabolic pathway shown here: glycolysis, the TCA cycle to produce energy. You also see that these reactions are irreversible. Pathways are irreversible, rather. So, you know, lipids are broken down to be put in these pathways. Intermediates of these pathways can be used to build up lipids and proteins and glucose as well. And so, as I mentioned in previous lectures, one of the favorite stars of biochemistry in terms of disease conditions that kind of encapsulates a lot of what we've talked about so far is phenylketonuria. So in that condition, we know that there's a defective enzyme, phenylalanine hydroxylase. So that enzyme converts phenylalanine, which is an essential amino acid, to tyrosine, which is non-essential. That's defective. So phenylalanine will undergo a transamination reaction, which we've talked about in this lecture, to produce 2 byproducts that are toxic to the brain. They're acidic, phenylpyruvate and phenoacetate. Those are toxic to the central nervous system and can cause mental retardation. So, of course, what we do when we have a baby that has been diagnosed with phenylketonuria, we make sure that they have a diet low in phenylalanine, high in tyrosine, so they circumvent that missing step or that missing enzyme, phenylalanine hydroxylase, and also we would ensure that they don't eat any foods that have phenylalanine, such as aspartame is one example that uses phenylalanine, that artificial sweetener. So again, this is just that pathway that kind of ties into a little bit of what we talked about, both with the defective enzymes, metabolism, and then transamination that we discussed in this block of instruction. So you guys are finished with the metabolic pathways. Do you guys have any questions about anything we've discussed in this block? Yes.
What is a carbon skeleton?
A carbon skeleton is a skeleton, in this case, without an amino group. Does that make sense? Everybody got that? And I saw another hand come up.
It was just gonna be about Diet Coke.
About who?
Diet Coke,
Diet Coke, yes.
you like it?
I love it. What else? That's it?
That's it.
Okay. What else? All right, I will see you guys at the review. Good luck on your A&P test tomorrow, Crayon. You got it. You guys will do it.